Good afternoon, ladies and gentlemen. Welcome to the ISDI virtual event titled Infant Formula and Adult Nutritionals, Analytical Methods and Current Challenges. We are pleased to have participants today varying from representatives of Codex member countries to colleagues from Codex observer organizations and ISDI members. My name is Marian Brestovansky. I am a Regulatory Affairs Manager at ISDI, and it is a pleasure to be your host today. Now I would like to introduce our speakers for the event. Mr. Darryl Sullivan is the chair of the AOAC Stakeholder Program on Infant Formula and Adult Nutritionals, or SPIFAN, and the Chief Scientific Officer at Eurofins Scientific. For Eurofins Scientific, Darryl provides expertise in designing comprehensive testing programs to meet scientific and regulatory requirements. He's an international expert in the analysis on infant formula and dietary supplements and has more than 35 years of experience in laboratory testing of food and dietary supplements. Darryl is considered an expert in the field of validation of analytical methods. He was a member of the task force that redesigned the AOAC standards development process and is the past president and the secretary of AOAC International Board of Directors. And as mentioned, he is also the current chair of the AOAC Stakeholder Panel on Infant Formula and Adult Nutritionals. Our second panelist, Mr. Erik Konings, is program manager at Nestlé Institute of Food Safety and Analytical Science in Lausanne, Switzerland. In his role, he provides leadership to global quality, laboratory, and regulatory teams to engage in strategic local activities to drive alignment and harmonization of analytical methods. He partners with government and non-governmental organizations in the development of standards for analytical methods. Erik is an active member of several standards developing organizations such as AOAC International, the International Organization for Standardization, or ISO, the European Committee for Standardization, CEN, and the International Dairy Federation, IDF. Both of our speakers today are active participants in the Codex Committee on Methods of Analysis and Sampling, or CCMAS, as part of the AOAC delegation. Today, our panelists will present SPIFAN, its goals, and its current work. They will provide us with an overview of analytical methods and challenges for infant formula and adult nutritionals. Darryl and Erik will explain the relevance of these methods in a wider context, their importance and value to Codex standards. We will also go through challenges in evaluating regulatory compliance for nutrients. Now I would like to give the floor to Darryl to go ahead and present the first part of the event.
Thank you very much, Marian, for that kind introduction, and thank you very much to ISDI and to Erik Konings for including me in this very informative webinar. I look forward to sharing some information with you today and hopefully answering all of your questions. As Marian said, I am the chair of the AOAC SPIFAN program and the expert review panel within AOAC. Today, I'd like to describe to you the AOAC process for SPIFAN, but not just the AOAC process, but the global impact that this activity has for standards and standards development for infant formula and adult nutritionals. This important topic has very widespread implications, and we're very excited to share this information with you today. So by way of background, the goals that the SPIFAN program had when we put it together were to update the AOAC official methods of analysis. But in addition to that, we wanted to engage global partners and harmonize standards that are used for testing of infant formula in many different parts of the world. And we also wanted to advocate global acceptance and use of these newly approved standard methods. So I'd like to share with you the process we use for these standards and how we go about identifying, prioritizing, and promulgating these very important standards. So the historical perspective of the SPIFAN program began back in the 1980s. The infant formula has been tested for many, many years, and AOAC had a very large number of standards that were validated and approved in the 1980s. While these were very, very good methods, in the last 30 years, there have been a number of new formulas with unique and novel ingredients that had identified gaps in the test methods that we had and caused some difficulties in the analysis of infant formula. Since infant formula is a highly regulated product, one of the most highly regulated products in the world, it was important that we had very, very good standards. So we began the SPIFAN program in 2010. Established with this acronym of SPIFAN stands for the Stakeholder Program on Infant Formula and Adult Nutritionals. We identified gaps in methods used to analyze labeled nutrients, since infant formula has to be labeled for up to 40 different nutrients, and every nutrient has to meet regulatory standards. The testing of these products and the evaluation of these products is highly critical. Here is a very nice timeline of all of these activities that have occurred in the last 11 years. And the details on this slide are not important, since I'll share some more information about each of these activities as we go through the presentation today. As you can see, this group meets frequently and has a lot of virtual and face-to-face activities that are important when you're discussing and identifying important standards and the important work that this community has been accomplishing. This is the organizational view of the SPIFAN process, administered by AOAC and having the standards development process with working groups and working group chairs, which I'll explain to you more in just a couple of minutes. Then we have the official methods process at AOAC, where we have first and final action methods that go through different steps of validation. Once methods have then been validated, they need to be controlled by a laboratory proficiency program, which then ties down all of the new methods and the use of new methods in various laboratories. So let's look at the SPIFAN community. Who's involved in this activity? If you look at the profile of stakeholders, you can see this is an industry-led activity, but industry cannot accomplish these goals by themselves. There are a lot of NGO organizations that assist with this. The participation from government is critical, and the academicians often provide significant technical support to our stakeholders. The specific perspectives are very broad across all sorts of different technologies. Essentially, anyone that's interested in the testing of infant formula is involved in this activity. So there's laboratories, manufacturers, standards and reference material providers, technology providers, regulators, and researchers all participate in the SPIFAN program. If you look at the international representation of our stakeholders, I believe there's as many as 70 countries represented in this slide that have all participated virtually, in person, by submitting comments or questions or helping us review standards as we work through this process. This is a very comprehensive list covering all of the geographies, and it's so important that we have this international participation in our standards activities. The process begins with the stakeholder panel and the advisory group that leads this activity. We then work on standard method performance requirements, which I'll define to you in more detail in just a moment. Once we have the requirements, we call for methods and look for experts to help us with these methods. We review the methods and decide whether or not we have sufficient methods to go through our SPIFAN process. So working through each of these steps, let's look at a little more detail of what this means and what this represents. The stakeholders are a broad representative group. The stakeholder meetings that AOAC holds are open meetings so that anyone that's interested in the analysis of infant formula is welcome to participate. You saw the very diverse group of people that participate in helping us identify what priorities are and what activities were going to occur. The stakeholders then identify what's important and what nutrients or analytes we'll be working on. When we choose a particular area of research, for example, vitamin D, we then form a technically focused working group. We select a chair who has expertise in the analysis of vitamin D, and then we collect experts from all over the world that help us identify what the important characteristics are, what the standard method performance requirements are, and how we proceed with the standards development process. The final step in reviewing these important methods is done by an expert review panel. Expert review panels are selected and identified based on expertise and experience for members that are interested in this activity, and they're vetted through the AOAC process and approved to be members of the expert review panel. This is a list of all of the program working groups within the AOAC SPIFAN process. I'd like to show this slide because you can see how comprehensive this list is, and I'd like to share the names of the working group chairs that are identified in each of these different groups. This is important because these individuals are experts in each of these areas of research and have been extremely useful in helping us identify the important characteristics of this analysis, and they continue to answer questions about the work AOAC is doing and about the different analytes that we're measuring and the criticality of each step and some of the challenges we have with each of these working groups. So the standard method performance requirements that I've been describing are a very detailed document that contain all of the necessary information for us to understand our test method. It begins with an applicability statement. What are we going to measure specifically? What forms of these nutrients? What types of samples will we be analyzing? And what sort of analysis are we expecting? The performance requirements include all of the things that chemists recognize that are important in a test method. The precision, the accuracy, the repeatability and reproducibility, the fitness for purpose of these methods as we look at their applicability to the analysis of infant formula. We also include any other details that are important with these standards as we look for each of the important aspects in the analysis of infant formula. Once we have decided on our recommendations for standard method performance requirements, we send those out for public comments. We answer every comment, every question that's submitted to us so that we're sure we have all the necessary information. We then publish these standards in the Journal of the AOAC for everyone to see. So here's all the priority nutrients, SMPRs, and methods that have been completed in the SPIFAN process. And as you can see in this table, it's very near completion, although we still have some gaps and things that we're currently working on, but we have made significant progress as we work through each of these steps. The final action methods for infant formula are listed in this slide. Each of these methods represent an official AOAC final action method, and they're codified, so these numbers are now imprinted in the AOAC process, and the numbers will never change. These are the references that are used by laboratories that are using these important standards and can be referenced as official analysis of infant formula through the AOAC process. So I've mentioned how important the global engagement is in this activity, and you can see in the center the SPIFAN process. But we also want to talk in this presentation about our engagement with Codex in how important that is. This couldn't be accomplished without our industry partners, without input from our regulators to make sure they're comfortable with everything that we're doing. And we have partners in ISO and IDF, the International Standards Organization and International Dairy Federation. And I'll talk a little bit more about that in a minute. AOAC has sections all over the world that have helped us with this process. And then specifically 2 geographies that we're working with in China and in India that have separate standards processes that we're working to help harmonize into the SPIFAN program. So in addition to all of the work that AOAC does, we have formed partnerships with ISO and with IDF. So each of the standards that we work on at AOAC are also reviewed and approved by experts in the ISO and IDF program, so that when we finish our research and validation, we have harmonized methods with AOAC, with ISO, and with IDF. We then have these standards that are promulgated, and you can see the AOAC methods that I mentioned earlier now partnered with the equivalent ISO IDF standard methods. So countries that prefer ISO references now have those that are harmonized in the SPIFAN process, and also the IDF reference that helps us understand each of these. Taking these standards now that have been validated by AOAC, by ISO, and by IDF, we submit those into the Codex process. The entry into this process is through the Nutrition Committee, CCNFSDU, and we submit these joint standards and propose that they be codified as Codex Type 2 dispute resolution methods. So then, in addition to the recognition by AOAC, by ISO, by IDF, we then gain recognition by Codex Alimentarius, which is recognized in 180 countries as the standards for dispute and testing of infant formula and adult nutritional products. These are the methods that I have shown previously with AOAC and ISO numbers. Now the highlighted methods are methods that have also been approved by Codex as Type 2 dispute resolution methods. Again, our chart is getting nearly completed, but we still have work to do. And I'll show you at the end of this presentation how you may want to help participate in this activity. I mentioned earlier that in addition to all the Codex countries and all the countries that represent and recognize AOAC and ISO methods, India and China have slightly different testing needs. In India, they have a separate system of official methods that have been used for many years for testing products in India. We have facilitated an agreement with FSSAI in India, which is their standards development organization, to recognize the AOAC methods that have completed the Codex evaluation. So those final methods now that have been reviewed by Codex are now entering into the process in India as well. We're also harmonizing with the China GB activities. The GB is a process that has Chinese standards that are used in country for the analysis of infant formula, and we're collaborating with scientists in China to begin looking at joint standards between SPIFAN and between the GB process. So we recently had our first method approved, a vitamin C method, and we hope we'll have many more chances for this activity. As I mentioned earlier, once you have completed the analysis of these important standards, use in the laboratory needs to be monitored through a proficiency program. So this is one example of a proficiency program that is administered by AOAC International that can help laboratories evaluate their performance with these standards and then apply for potential accreditation using these SPIFAN methods in and around their laboratories. So this is a very comprehensive list of all of the activities that have been accomplished by the SPIFAN program. We have over 50 first action AOAC methods, and we have 25 final action methods that have completed both the single lab validation and the multi-lab validation process. We have a number of working groups still active, including A2, beta-casein, and phospholipids, and we're beginning a new working group on SN2 fatty acids. We have 22 methods that have been approved jointly by AOAC, ISO, and IDF, and we have 15 standards that have been approved by Codex as type 2 dispute resolution methods. This activity has engaged more than 60 countries to help us in this review and harmonization process, and we hope to continue this process and expand the activities as we move into the future. So currently we are looking for experts that can help us with SN2 fatty acids. We're looking for individuals or laboratories with expertise in human milk oligosaccharides, lactoferrin, GOS, fluorides, and carotenoids. And we're looking for laboratories that are interested in participating, not only in proficiency, but in validation studies. If you have an interest in learning more about this activity, participating in learning, or potentially becoming active in the SPIFAN process. So all of these activities are made possible by our infant formula partners. They contribute financially to our success, and they contribute significantly to the expertise that allow us to promulgate these standards, validate these important methods, and then work through our process in the approval and evaluation of these important standards. So I hope you have a number of questions that you can share, and I believe we'll be entertaining questions at the end of our session.
Thank you very much, Darryl. Thank you very much for your presentation. And indeed, the participants can submit their questions through the Q&A, and we will take them in the end of the event. So if you have any questions, please type them in the Q&A function. So now I would like to move on to Erik, who will take us through challenges in evaluating regulatory compliance for nutrients in infant formula. Erik, please, you have the floor.
Thank you, Marian. Thanks for the nice introduction. Thanks to ISDI to giving us the opportunity to elaborate on SPIFAN and all the work. Thanks to Darryl for explaining on it. And despite the enormous amount of work which has been done in the development of new standards, we have still some challenges. Today, let's see if I can move. Yeah. Today, I would like to discuss with you three of them. Some of the current state-of-the-art analytical methods are not able to generate results within the regulatory limits. The reason for that is that the variability of the methods are still too high as compared to the regulatory limits. So as you can imagine, when this occurs, it is possible that a product which is truly compliant with the regulatory limits and falls within the regulatory limits is identified as non-compliant because of this issue. I will elaborate on this and specifically give an example on the situation for the vitamin D and the regulations in the European Union. Secondly, you have also seen Darryl presented on the huge list of the nutrient panel for which new standards have been developed. And we move currently to a new set of ingredients which are also added to infant formula. However, they have no provisions in the Codex 72 standard as of yet. This provides some challenges as well. And thirdly and lastly, we have continuing challenge because of the absence of harmonized definitions for nutrients, which can lead to potential disputes while verifying regulatory compliance. Infant formula producers must follow good manufacturing practices with strict process controls to ensure products are adequate, safe, and fully comply with regulatory requirements. Regulatory limits based on safety and nutritional considerations, as well as quality and functional properties of the product, must be taken into account. Also, regulatory requirements must be specifically met for each country in which the product is marketed. This can be quite challenging when a single product is sold in multiple countries with diverging regulations. To ensure products meet the highest standards, the global infant formula industry works to ensure a thorough understanding of the capability of an analytical method. That is the ability of a method to produce test results within the regulatory limits as an additional key parameter when establishing compositional criteria for nutrients in infant formulas. And we performed a study on this recently by evaluating the impact of the analytical method variability on the assessment of product compliance with the applicable regulations. The scope of the method included an estimation if the current regulatory requirements for infant formula, including the minimum maximum tolerances, can be enforced with these new AOAC ISO IDF SPIFAN methods. Within the scope were regulations for infant formula, foods for special medical purposes for infants, follow-up formula, and you see also we apply this for a range of nutrients and as well to countries which are representing regions with the highest infant formula sales. There are 4 steps to evaluate method variability on the assessment of product compliance. First, the determination of product specifications based on the national regulations. Then we had to review the analytical method variability of the existing AOAC ISO IDF methods. We defined a method capability, and we evaluated the results of this method capability and the ability of a method to demonstrate compliance with the regulations. We acknowledge that all analytical test methods have some uncertainty. The key factor is understanding a method variability in the context of the overall variability, which also includes variation in raw materials and ingredients, and also the manufacturing process. The overall variability must fit within the regulatory limits as illustrated in this figure. You can imagine here the analytical variability is this, if this becomes too high as compared to the regulatory limits, the lower and upper regulatory limits, then the method produces test results outside these limits, whereas in the true value of the product is in the middle of the regulations. Analytical method variability values from the current AOAC ISO IDF methods were taken as these methods are currently considered best in class for measuring nutrients in infant formulas and adult nutritionals with superior precision and trueness to any methods to date. To represent method variability independently from the laboratory using it, we took the RSD big R values, or the reproducibility values of the methods, as estimates in our study. Six Sigma is an initiative for continuous improvement developed by Motorola engineer Bill Smith in the mid-80s. In this approach, Sigma, used by statisticians to denote SD, can be considered as a process variability and a concept representing the amount of variability relative to requirements of specifications. From this concept, the impact of analytical method variability on the assessment of product compliance can be assessed for a given analytical method by defining a method capability index, CM. The method capability index compares the specifications for a given parameter to 6 times the estimated method variability. The variance of the analytical method is taken as 1/3 of the overall process variance, considering the other 2/3 coming from raw materials, ingredients, and the production process, which I've shown to you earlier. Reworking this information results in a method capability lower than 1.73, which will become a concern, and a method capability lower than 1 becomes critical, because there is a risk of obtaining analytical results outside the specification range, even though the product is truly compliant with the product specifications. What the impact is of the method capability lower than 1 with regard to compliance to regulations, I would like to explain using the example related to the vitamin D regulation in the European Union. You see with the red lines the upper and lower regulatory limits. You see also with the 95 confidence interval, which is generated for an analytical result using the current standard, there is a risk that the probability to find a test result outside the regulatory requirements is 90%, even though the true value is in the middle of the regulatory requirement. So this means that 1 out of 5 tested products is identified as non-compliant, although compliant. So the method variability needed, you see here, so that you will not generate results outside the upper and lower regulatory limit. It needs to be 2%. And actually for vitamin D in infant formula, that is impossible with the current technology. As mentioned earlier, the current standard is best in class with 8% and far from this 2%, which is really needed to test compliance with the regulatory limits. Conclusions from this part. Some international methods are not fit to demonstrate compliance with the narrow nutritional regulatory limits of several countries. The method capability is below 1. The probability of finding a test result outside the regulatory requirements due to analytical variability alone can be as high as 90%. So as you have seen in the example of vitamin D with European regulatory limits. Of particular concern, and I showed vitamin D, but there are some other methods, for example, vitamin A, B12, and folic acid for country regional regulations in China, European Union, Pakistan, Russia, Singapore, South Africa, and Thailand, and particularly for vitamin B1 and B2 in FSMP pertaining to the Russian regulation. The situation could be improved by new analytical methods with less variability, if this is at all possible. And as already mentioned, the current standards are state of the art, and we think not possible with the current technology. It's also possible to change, harmonize regulatory limits to fit testing capabilities. And we have seen, I have not included in this presentation, but the several regulatory requirements in several parts of the world are very different. And harmonizing this would make sense, and also include the technology, what is possible to test compliance to these regulations. To set regulatory limits, it could be worth to implement the concept of method capability to set and evaluate performance requirements supporting the assessment of regulatory compliance. If you would like to know more details about this study and to have more explanations on how it was done and the outcome, I would like to refer to a recent publication in Food Control published at the beginning of this year. And we joined efforts among several infant formula manufacturers for this publication. I would like to mention the second challenge, the Codex recommended methods, for which there are no provisions in the Codex Standards. We have non-recommendations. There are currently some discussions on this aspect. I would like to take an example of fructans. Fructans is their carbohydrate polymers composed predominantly of fructose monomers. They are added to infant formula for their prebiotic effects. And within the SPIFAN program, we have established AOAC ISO IDF standards to quantify fructans in infant formula. There are already other standards existing in the world to determine fructans in food. However, these standards are not fit for purpose to analyze the low levels of fructans in infant formula. So we need specific standards to verify compliance to claims or labeling. Actually, CCNFSDU has proposed last year to CCMAS to review these methods for adoption in the Codex system. However, CCMAS has decided to send back this proposal to CCNFSDU to let them clearly explain what is the rationale for including Codex recommended methods for nutrients, ingredients for which there are no provisions yet. We think that considering the language in the Codex Standard and the current Codex Standard 72, there is a rationale to include Codex recommended methods in Codex Standard 234 on recommended methods of analysis and sampling, because this standard is stating infant formula is a product based on milk of cows or other animals, or a mixture thereof and or other ingredients which have been proven to be suitable for infant feeding. So fructans is one of the example which have been proven is already largely applied and added to infant formula. Furthermore, some authorities in the world, they request to identify and use suitable internationally recognized methods for product registration. So for product registration in certain countries, you have to recommend such a method. And it would help if we can refer to recognized internationally accepted methods, and in this case, Codex adopted. Not only fructans is one example. Currently, within the SPIFAN program, and Darryl mentioned it already, we are working, and there are some new standards in the pipeline for other new ingredients like HMO, human milk oligosaccharides, galacto-oligosaccharides, lactoferrin, lycopene, A2 beta-casein, and fatty acids in the SN2 position of triacylglycerol molecules. The last challenge I would like to mention is still we have challenges in verifying regulatory requirements, regulatory compliance, because of the absence of harmonized definitions of nutrients, and it applies to nutrients in general, but today I would like to take an example of vitamin A. In the Codex Standard 72, vitamin A is specified as all trans retinol. It further mentions that carotenoids should not be included in the calculation of and declaration of vitamin A activity. Another Codex guideline mentions, and it is about an advisory list of nutrient compounds which are allowed to be used in infant formula, allows all trans retinol, retinyl acetate, and retinyl palmitate as ingredients to be used in infant formula. Actually, the current vitamin A activity scientifically is seen as all trans retinol, and it should include another form, the 13-cis retinol. This compound, 13-cis retinol, is known to have 75% relative to all trans retinol as a bioactivity, and should be included if you measure total vitamin A activity. Infant formulas may contain up to 25% of 13-cis retinol, depending on the processing conditions. So coming from the vitamin A sources allowed to add it in infant formula. As we look to the SPIFAN process before establishing official methods, Darryl mentioned already briefly on it, we identify the standard method performance requirements. And an important element of this is the so-called applicability statement. And for that, we clearly establish with stakeholders what do we mean with vitamin A, and we do that for every nutrient. So we identified that a standard official method for vitamin A in infant formula should be based and should measure all trans retinol and 13-cis retinol. So the method does that as well, and also other methods like, for example, a SEN method, a SEN standard for vitamin A in foods also includes 13-cis retinol and all trans retinol. You can imagine if there is no clarity about what needs to be included in your methods, you can add up with disputes with laboratories having or measuring different compounds, different if they have another understanding of what should be measured as vitamin A. I think in the past, CC and FSDU in 2016 had a discussion on vitamin E, and they defined particularly for vitamin E, this nutrient, to focus on alpha tocopherol for labeling purposes. So there is a few discussion in this case, vitamin A. I give the example of vitamin A, but these discussions apply also to a number of other nutrients. So for us, a challenge while we progress in defining and using official methods to verify regulatory compliance. As a summary, the analytical method variability is considered as one of the three main sources of overall process variability, which also includes the variation in the raw materials, ingredients, and the manufacturing process. Important element. The concept of method capability is an adequate tool to help set or evaluate performance requirements of future methods to deliver results which support the assessment of regulatory compliance. Codex recommended methods for nutrients without a provision in CXS 72 and infant formula can help in the alignment of verification of label claims for new ingredients as well. Harmonized definitions for nutrients would help to align official methods in the quantification of nutrients and avoid disputes on the verification of regulatory compliance. And with that, I would like to thank you for your attention and give back the floor to Marian.
Thank you very much. Erik, thanks a lot for your presentation and for both of your presentations. We have received some questions in the Q&A, and I also do have some on both of you. So if you permit, I will ask some questions to you. So first, Darryl, so first of all, congratulations on all the work of SPIFAN, and thank you for presenting it and for taking us through the activities. You mentioned it in your presentation, and you took us through how the methods are developed. But I was wondering how long generally does it take for a method to be developed or updated in SPIFAN before it is brought to Codex?
Thank you, Marian. That's a very good question. So in my presentation, I showed all of the different steps that are needed in the promulgation and validation of these standards. And the laboratory activities take some time to go through a single lab validation and a multi-lab validation. But at each step of this process, we also need to gather stakeholder input. So we post each of our recommendations for a minimum of 60 to 90 days so that we get input from all of our international stakeholders that may or may not be able to participate in person. So there is quite a range, but in general, it will take from 3 to 6 months to complete the first step of the AOAC process, up to 1 year to complete the AOAC process, and then generally another year for the Codex process. So in general, from 1 to 2 years from beginning to end through the Codex process. We have done things very quickly when there's been an emergency, but in a general sense, that's about how long it takes.
Thank you very much. Thanks. Erik, I also do have a question for you. And you also talked about this in your presentation, and you talked about the probability to find a test result outside the regulatory requirements for vitamin D, and that it's at 19%. So I was wondering, what are the factors contributing to such a probability outside regulatory requirements, and is the probability, how higher is the probability for vitamin D than for some other nutrients?
Thank you, Marian. Yes, it depends on several factors. First of all, you have the variability of a method. The method process, technology used, this is one. You have certain concentrations of nutrients in your infant formula. So particularly for the nutrients I mentioned, vitamin D, vitamin B12, vitamin A, they are very low in amounts, in absolute amounts in the infant formula. So that means a higher, let's say, chance that there is a variation with the technology used. Then a third element is also the regulatory requirements as opposed to this variation of the method. So we have seen in vitamin D it's a quite tight regulatory requirements. So it is not possible to, with the current technology, to estimate vitamin D in such low levels within these tight, let's say, requirements of the regulation. This is a higher chance and gives a higher probability of methods to find value outside these regulatory requirements. Then, for example, we have seen for minerals, which uses a technology which is quite higher in concentration, and there it is not such an issue as compared to these vitamins I mentioned. So I hope this I clarified a bit on that. So yeah, a combination, and therefore, as I mentioned, the regulatory requirements are not the same in every part of the world. So for vitamin D, the method as we have it, and I demonstrated you the variation fits within the regulatory requirements, but particularly in this case in Europe, quite tight regulations, it becomes a challenge. And maybe, yeah, the requirements in some part of the world are different, and that means that maybe to set these limits, there are other risk assessment procedures to set these limits, and this might be another discussion to take. But maybe it's good to also consider this in the future, and first of all to consider these setting of these limits. But while setting these limits, it should also be considered what the current technology is available to do to verify these regulatory requirements, because also within Codex procedure, it is a requirement for provisions to have a suitable method in place to verify these requirements. So important element for to consider in the future.
Thank you very much. That's clear. It seems quite complex, and it seems that indeed the different regulatory environments make it sometimes more complex, but thanks a lot for that explanation. Darryl, I believe I do have a question for you from the audience. There was a question whether you could explain again maybe the process that is followed in India and China to develop methods, and how do they follow different processes basically than international?
That's a very good question. Thank you. So most of the SPIFAN activities have occurred in the US just because of availability and because of activities going on in the US. There has been limitations in the participation from India and from China, both because of the geographical distance, making travel difficult, and because of the time differences, trying to communicate remotely. So AOAC has sections in both India and China. They're like separate chapters of AOAC. So we have worked through the experts we have in those different sections and engaged with the regulators and with the industry in both India and China. We have traveled before the pandemic to India and China with our SPIFAN experts and held in-person meetings in both India and China to help them understand the process. So in India, we're working with the regulatory agency FSSAI that promulgates standards in India for the analysis of infant formula, and we're working with the GB standards development process in China to help harmonize the standards activities in China and in India with the AOAC standards process. This is a slow process, but we continue to make advances with each step, and we will continue this activity into the future as we do more and more validation studies.
Thank you very much. Thanks a lot for that answer as well. And we have one more question, and we do have time for one more question. Maybe Erik, there was a question from the audience whether there is a standard process when a result is out of critical limits due to variability of the method, just one additional analysis, or is this the case minimum number of additional results needed?
Thank you for the question. I would think, I did not talk about it today, but measurement uncertainty is a general approach that labs using the method should take. And this uncertainty gives already a good view on the variability of the method and within what limitations the method can produce. Furthermore, it's up to the, let's say, the laboratory, how they define this up to one, or they can do duplicate or triplicate analysis, even if they find a result outside the regulations. In my example, I took the assumption that the the manufacturer, the product was manufactured exactly at the regulatory mean. But okay, this can vary, of course, in the process. So yeah, first of all, I would say take a look at the measurement uncertainty processes while analyzing products, and define on that when a result is found outside the regulations, to minimum duplicate your test to be sure as well. Thank you.
Thank you very much, Erik. Thanks a lot also to the audience for the engagement and for your questions submitted through the Q&A. If you have any further questions, you can contact the ISDI Secretariat after the event, and we can connect you with the speakers. So again, I would like to thank Darryl and Erik for their time today. In this event, we have learned about SPIFAN, elaborated about its activities and processes to develop methods. Darryl emphasized the global coverage of SPIFAN and mentioned the experts that are key to its success. We also learned about the submission of analytical methods to Codex and to Codex committees, and the process that surrounds this exercise. The audience also heard about challenges in evaluating regulatory compliance for certain nutrients from Erik. Erik also talked about analytical methods variability and the concept of method capability. And he also highlighted that harmonized definitions for nutrients would help to align official methods and avoid disputes on the verification of regulatory compliance as one of the key points. Again, I would like to thank our panelists for their interventions, and I hope that the participants found today's event interesting. It certainly seemed so, looking at the questions and engagement from the participants. So finally, when leaving the webinar, you'll be invited to fill in a 1-minute poll where you can provide feedback on today's events. So please go ahead and submit some of your comments on the event. And again, thanks you all for your attendance, and I wish you a great day.